Rothman lab at UCSB, and I just want to start by acknowledging that none of this research would have been possible without my graduate student Caroline Ackley, so I want to thank her for letting me join. And today I'm going to be talking about my research on C. elegans, studying their mechanosensory behavior, negative gravitaxis, and I know that sounds insane, but to put it simply, I just study these worms' ability to sense gravity. And so to start, I just want to explain what are C. elegans. Yeah, so they're a common model organism used in scientific research. And so specifically, they're microscopic roundworms found in the soil across the globe. And so they're a really good model organism to use because they're so simple. And so they only have 302 neurons, which might sound like a lot, but humans have around 86 billion. So yeah, it's a lot simpler. And all of their neuronal connections are fully mapped, making them a really good organism for scientists to use. And so one of the first questions I often receive in my research is, why should I care? How does studying a tiny little worm impact humans and the way we sense gravity? And so a common anecdote I like to explain in scenarios like this is, if you've ever heard of circadian rhythms, the genetics underlying circadian rhythms was actually first discovered in the fruit fly. And so much in the same way, I hope my research studying the gravity sensation abilities of these C. elegans will later be able to translate into humans. And so humans do actually constantly sense gravity. We do this through something called our vestibular system present in our inner ear. And so we have these little rocks called otoliths. They're made of calcium deposits, and they shake around basically on our inner ear present on a sheet of sensory cells. And so the weight of these rocks is pulled down with gravity, and as we move our head from side to side, that's how we orient ourselves and our sense of balance. And so we are aware that this happens in humans, but my graduate student was shocked that she discovered these C. elegans might be detecting gravity themselves. And the real reason that this is so amazing is they don't have a vestibular system. They're incredibly simple organisms, and as I said, they only have 302 neurons. And so how do I actually test that they're sensing gravity? How am I aware that this is what they're doing? Well, I create this experimental rig where I essentially take two tubes and I put them on top of one another, and I fill them up with a gel. And then I inject a dense pellet of these worms into the center, and then after 24 hours, I plot their distribution, either traveling upwards or downwards. And so this is just what the quantified results look like for a basic control. And as you can see, there's a very clear upward distribution. So they're clearly sensing gravity to some degree and traveling upwards. And so this sounds super fancy, but we've deemed this behavior something called negative gravitaxis, because they're negatively sensing gravity, and instead of going downwards, they're going upwards. And so you might be asking yourself, well, how do I know that they're sensing gravity? There's a lot of different sensory inputs that they could be receiving. And we know this because we use something called a Faraday cage. And so Faraday cage, they block Earth's electromagnetic forces. And we specifically with this Faraday cage also had it so it blocks light, and we've measured it so there's no strict temperature gradient from the floor to the top of the cage. And so with all of these factors combined, we've kind of eliminated all of the possible sensory inputs except for gravity sensation. And so this was originally somewhat disputed in the field because these organisms are so small and simple. It wasn't thought that it was physically possible for them to be able to sense gravity, but as I showed you with the quantified results, they have a very clear upward distribution. And so once we discovered in the lab that this was possible and this is what they were doing, now comes the interesting part of my research, which I'm really interested in, and it's how are they possibly doing this? And so I specifically studied this set of neurons called PVD neurons, or polymodal ventral dendritic neurons. And so they're present along the entire body of the C. elegans, and they have this very clear morphological shape. And so we found that mutating these specific neurons, which are known for thermosensation and pain sensation, completely eliminated the behavior. We no longer saw that they moved upwards and downwards. And so this alone was a pretty incredible feat. I was really excited because, as I said, there's no structural component to the C. elegans that makes it responsible for this behavior. So it has to be something present within their neural circuitry. And so even beyond this, we started studying what specifically within these neurons is responsible for this behavior. And this is the part that I thought was really cool. We found these TRPA1 ion channels. And so essentially an ion channel is a pore-like channel present in your neurons that allows the neurons to do a bunch of different things like secondary mechanism systems and allows for action potentials. And so we found that specifically mutating these microscopic channels within the neurons of these microscopic organisms also change their behavior. And so I've studied many different strains, but specifically 2 different mutants of these TRPA1 channel mutants. And so the first strain I studied completely eliminated the negative gravitaxis distribution. As you can see here, they had a more common distribution of the bell curve with their movement rather than a clear distribution upwards. And then one of my most exciting results from my research occurred when we found an even more strongly mutated version of this channel. And so with this stronger mutation, the first couple times I started scoring these experiments, I thought I was going insane because every time I did the results, I found this really weird phenotype where they were all traveling downwards. And I had never seen that before in the lab, no one else had, and so I thought I must have been doing something wrong. But then as I kept repeating this experiment over 7 times, I realized that we had somehow reversed the behavior. And so this is going to be the focus of my research for the future years, and I'm really interested in how this specific ion channel can possibly play such a big scale role in this research. And I want to thank everyone so much for listening, and I hope if you have any main takeaways from my research is that the next time you see a little insect running across your counter, you realize that they are interacting with the world in incredibly complex ways, more complex than we can ever imagine. And beyond this, studying the way that they view the world can actually help with understanding the human body as well. Thank you so much.
So the mutations that you studied, do you know exactly what happened to the protein? Like how was the protein disrupted?
I think the pore section of the mutation caused the pore of the channel to be displaced, so there wasn't that clear ion distribution. And in one, it was like a frame shift mutation that had an early stop codon, and it completely disrupted the channel. But in the first one, where we still saw the behavior, ions could still pass through, but it was less easy for them to. Yeah. Yes?
So do you think the actual gravity detection is based on just that ion channel, or is it interacting with the passage of the signal?
So right now we're not totally sure, but I definitely think the ion channel plays a role in the way the neuron senses this gravity, and it's definitely present in the entire body of the organism because, as I showed, the neuron is within the entire length. But I also think now that there might be something to do with iron deposits, and so there's a lot more research to be done in this section. Yes?
It looked like for the sample that went down, they didn't go down as far as the ones that walked up. Is there a reason why they didn't go all the way down to the bottom of the sample, whereas other ones, there was a lot that walked all the way to the top?
Yeah, so mainly we found that they didn't always travel to the extremes of the tube, and so overall, there was less of that clear distribution, but yeah, because of their gravity sensation capabilities, they usually went up as far as they could.